One of the fundamental challenges in modern biology is to understand the operating manual of a living cell, not just identifying its components, but mapping the causal relationships that govern its behavior. In this explainer, we'll examine how causal machine learning is providing a new framework to decode this biological blueprint.
You see, standard machine learning methods are very effective at identifying patterns. For instance, they can observe that when the sun is out, sales of both ice cream and sunscreen increase, but they don't inherently understand that the sun causes both events. In biology, this limitation is significant. A model based only on correlation might fail when predicting the effects of a new intervention like a drug. Causal learning aims to go a level deeper, to discover the underlying mechanisms, the actual wiring that drives cellular function.
So, here is our agenda. We'll begin by discussing the limits of correlation. Then we will look at how biological cause is modeled. From there, we'll explore three primary challenges for the field: predicting novel outcomes, interpreting the models, and capturing cellular dynamics over time. And finally, we'll consider the path forward.
Let's start with this core issue. Why is it insufficient to rely on patterns and correlations alone?
This table outlines the distinction. Traditional statistical learning excels at prediction as long as the conditions remain the same. It identifies correlations in the data it was trained on. Causal learning, on the other hand, aims to discover the underlying mechanisms. The goal is to build a model that understands why a system behaves the way it does, which should allow it to generalize and make predictions about new conditions it has never encountered.
So how can we formally model this web of cause and effect? The primary framework used in this field is known as the structural causal model, or SCM.
An SCM can be thought of as a wiring diagram of the cell. In this diagram, each node represents a variable, like the expression level of a gene. A directed edge, an arrow, from one node to another signifies a direct causal link. An important constraint is that the graph must be acyclic, meaning it contains no loops. This ensures that a variable cannot, through a chain of events, end up causing itself.
This graph here represents a cell's baseline state. It's the map of its default gene regulatory network, where the expression of certain genes is determined by their parent genes. This serves as our foundational blueprint before any experimental intervention is introduced.
Now, when an experiment is performed, we can model it as an intervention on this graph. The most idealized form is a perfect intervention. This would be like a flawless gene knockout, where we completely sever the causal links from parent genes to a target gene. The target gene's activity is then set to a new value, regardless of the state of its former regulators.
Of course, real-world biology is more complex. Interventions are rarely perfect. A CRISPR-based method might have off-target effects, or a drug could influence multiple pathways simultaneously. These imperfect interventions are more difficult to model because they introduce multiple changes to the underlying causal graph.
This brings us to the first major challenge: building models that can accurately predict the outcomes of conditions they haven't been trained on.
This is the essential question. The ability to forecast a cell's response to a novel compound or a new combination of genetic perturbations would be a significant advance. It requires a model that has learned the underlying causal rules of the system, not just the correlations present in its training data.
So what makes this so difficult? There are several practical hurdles. First, building these models requires large and diverse datasets of high-quality interventional data. Many existing datasets provide only a limited number of cells per experiment. Furthermore, technical noise and batch effects can obscure the true biological signals. And often, we are measuring proxies, like RNA abundance, rather than the direct protein activity we are truly interested in.
The second challenge is interpretability. A model that makes accurate predictions is useful. A model that can also explain the biological reasoning behind those predictions is far more valuable.
This is the key. The goal is to avoid creating a black box, a system that provides correct answers without revealing its internal logic. For these models to be truly useful to scientists, we need to be able to inspect their structure and extract new testable hypotheses about biological mechanisms.
The scale of this challenge is immense. To put it in perspective, consider this number, 100 billion. This is a rough estimate of the number of possible interactions between different forms of proteins, or proteoforms, within a single human cell. A model must navigate this vast combinatorial space, which makes simple interpretation quite difficult. To manage this complexity, models often use what are called latent variables. These are internal, learned representations that can capture unobserved factors, like the activation state of a signaling pathway. While these variables are powerful for prediction, they pose an interpretation challenge. It is not always straightforward to map a model's learned latent variable back to a known biological concept.
The third significant challenge is incorporating the dimension of time. Many critical biological processes, such as cellular differentiation, are dynamic and evolve over a temporal course.
A cell is not a static entity. It is a dynamic system. It develops, differentiates, and responds to stimuli. A single snapshot in time often fails to capture the full picture. The challenge, then, is to develop models that can represent these entire cellular trajectories. A primary obstacle here is that many single-cell measurement techniques are destructive. We can't continuously monitor a single cell over time. A common computational solution is the concept of pseudotime. The process involves taking a snapshot of thousands of cells at once with the assumption that they represent different stages of an ongoing process. An algorithm then orders these cells along a trajectory from start to finish to reconstruct a synthetic timeline. This provides a powerful way to study temporal dynamics from static data.
So considering these challenges, what is the path forward for causal modeling in biology?
Progress will likely require advancements on two fronts. On the experimental side, there's a need for more reliable interventional data, more time series observations, and greater standardization of protocols across different labs to improve data quality. On the computational side, continued development of more effective and scalable methods is needed to analyze this increasingly complex data.
The ultimate vision is a feedback loop between computation and experimentation. Causal models could be used not just for prediction, but to guide the design of future experiments. A scientist could query the model to identify the most informative intervention to perform next. This approach could substantially accelerate the pace of discovery and the development of new therapies.
To quote from the source material, causal models hold the potential to help scientists navigate the vast complexity of biological systems by uncovering novel insights and accelerating the discovery of new therapeutic interventions with greater precision and efficacy. This summarizes the potential impact of this research area quite well. And this leaves us with a forward-looking question. The long-term ambition for this field is, in essence, the creation of a predictive in silico cell. As our datasets grow and our models become more sophisticated, the possibility of building a digital twin of a cell, one that could be used to simulate any number of interventions, becomes a more tangible, though still distant, goal.